Okay, so welcome back. So we'll get started from where we left in the previous video. If you remember, we were looking at the solvent extraction. So we were basically looking at this particular topic, recovery of precious metal from solutions by solvent extraction. We looked at different types of solvent extraction for gold. We also talked about things in the presence of ammonia, without ammonia. If you remember, we looked at those two big equations, and then we will continue that discussion in this particular video as well, where we will look at some of the other way of extracting these heavy metals, these precious metals from the electronic waste. So let's look at the next aspect. Like I say, we also use activated carbon. So activated carbon is has been this is if you look at the literature in terms of trying to extract gold from either the ore of the gold or also from electronic waste. There is a people have looked at the mechanism, people have looked at the kinetic, and as you know, the activated carbon, I assume that most of you have some sort of environmental background. So you will, if you have taken the water class, or even in some extent in the wastewater, if you have gone all the way to the tertiary treatment, we do use activated carbon. Activated carbon is essentially adsorption site. So as you can see, this is a, and there is a difference between adsorption and absorption. Adsorption is a physical phenomena, and absorption is essentially you have some sort of a physical as well as a chemical phenomena can take place over there. So adsorption, so you have a layer. So if you think about, if you look at, say, this is a surface, I have this surface, and on that surface things are coming and attaching it to it. So more is the surface area, bigger the particle, like a surface area, smaller the particle, more the surface area, better is the adsorption. So that's why activated carbon usually have very high surface area. So here what we are trying to see is that how the gold is getting into this activated carbon has been looked into by many researchers, and they look at the adsorption process. And whenever we talk about adsorption process, we have the concept of the isotherms comes in, like a Freundlich isotherms or Langmuir isotherms, and you have a partition coefficient number. Partition coefficient is how much would be in the solid phase versus how much would be in the liquid phase. So for the solid and liquid adsorption process, this is the typical equation that is used. So we can this is the typical relationship where Q is the quantity of solute adsorbed per unit weight of carbon. So this is what is getting into the, this is the solute which is getting adsorbed onto the carbon. Q max is the maximum sorption capacity at the saturation. C is the equilibrium concentration of the adsorbate. Adsorbate is the what is getting adsorbed in solution, and B is the adsorption equilibrium constant. So there is a that's your equilibrium constant. So based on that, you can write down this relationship where Q, which is the quantity of solute adsorbed per unit weight of carbon, is equal to Q max, which is basically the maximum sorption capacity, multiplied by your B, which is the equilibrium constant, C is the equilibrium concentration of adsorbate in solution. So you can write down this same equation as a typical equilibrium equation that you have done in a chemical equilibrium class, and then you can rearrange that and you can get that same equation there as well. So activated carbon is also used in terms of getting gold from solution. So we are basically trying to remember from the previous class what we are trying to, what I was trying to tell you is that you start with we have this electronic waste, or for that matter iron ore. From the solid phase, first we try to bring it to the liquid phase, and from the liquid phase again we try to bring it to the pure solid phase. So that's the phenomena. We already talked about how to get it from the solid phase, from the electronic waste, or from the ore to the liquid phase. Now we are talking about how from that concentrated liquid which has gold present or this precious metal present, how we'll recover this precious metal. So that we are trying to look at. So another expression that is used for these kind of activated carbon or the isotherms is the Freundlich isotherm, which is very common. You may have seen it in other classes as well, where you have Q is expressed as K times C to the power of n, where your K is K and n are the Freundlich constants, and C is the concentration. and Q is C is the like a maximum concentration, and Q is the concentration at like at a particular when you are looking at how this is the maximum like based on maximum sorption capacity, and the Q is the sorption capacity at particular for that particular like material. So, K and N will be Freundlich constant for based on the activated carbon. So, Freundlich type multi-component isotherm was developed by this another things are developed. So, again what I am you do not have to kind of memorize or these things. What we are trying to say over here is there are a lot of work going on. There are a lot of work going on in terms of looking at the recovery of gold from electronic waste and to bring the pure gold. There is still a lot of work, mostly in the lab scale, some pilot scale. Full scale plant of recovery of gold, there are some in Japan, some in Europe, but we don't have any, as far as I know, in India, where we have this process, the entire process, and that's where a lot of research is needed in the country. So, those of you who are looking at electronic waste as potential carrier for your master's or PhD project, master's or PhD thesis, these things, and you are interested in chemistry, you have to be interested in chemistry, and then you can pursue these kind of topics. Ion exchange, so all these are nothing new, like adsorption, activated carbon, ion exchange, even the previous the leaching, thiosulfate leaching, cyanide leaching, all these have been used in different fields. So, we are trying to just apply that knowledge with electronic waste, recovery of precious metals from electronic waste. So, in terms of the ion exchange, commercial resins are used small like along employing a small quaternary ammonium it's called ammonium moites, a strong base resin, and that's have been found that once you have this thiosulfate leaching, remember we talked about the thiosulfate leaching. Now, this thiosulfate has gold there, so we can use this resin to recover the gold from this thiosulfate liquor. So, this particular research, they observed that almost full recovery of gold and silver using a strong base resin. So, they could recover gold and silver from there. Aqua regia leaching can be used as well, and where aqua regia is used. Aqua regia is used mostly in the informal sector when they try to do this gold recovery. They use a lot of aqua regia, and that is used in the recovering of gold from computer chips due to its flexibility, ease, and low capital requirement. So, that also has been found. Has been reported in the literature. So, these are some of the ways in which you can extract gold from the liquid phase. So, and for just to have a process, how this process would look like if you want to recover metals from PCBs. Now, what is PCB? This PCB is not that polycyclic biphenyl that mostly the PCBs that we hear and talk about in the environmental field. Here this PCB is the printed circuit board. So, when we say PCB, actually we are looking at the printed circuit board here, not that PCB that you see in your most of the environmental book in terms of polycyclic like biphenyl. So, PCB scrap, which is the printed circuit board, which is there from can be from computers, can be from any sources like any electronics, wherever you have some sort of electronics, it will have a printed circuit board. Even your cell phone, you open, you will find a circuit board. So, there is a circuit board. So, how will you how we potentially go in terms of recovery of metals? First thing, we try to do the shredding. Again, why we do the shredding? I keep on telling you, maybe many times I have said even in this 4-week class so far, that everything that you look at, try to understand why it is being done. Why is shredding? Because shredding, that means extra energy source, isn't it? You are applying, at one point we are talking about environmental management, environmental protection. So, if shredding is not, we have to understand why the shredding is required, because shredding means we are setting up a separate unit. Shredder has to be set up. We are talking about printed circuit board. So, they are not easy to shred. So, it will require certain energy to do that, and then energy means environmental footprint. So, we have to understand why the shredding is required. I think by this time I should you should be able to tell me why it is done. It is essentially what we are trying to do. By shredding, we are reducing the particle size, increasing the surface area, isn't it? So, we are making it a small particle, we are making it a should not call it particle, like a small pieces. small pieces of printed wire board, and small pieces means more the surface area. More the surface area means better the reaction, and that is why you are trying to do the shredding. So, after shredding, you can do the leaching. Now, what we are trying to what essentially we are trying to do in leaching, we have this we have these heavy metals, the heavy metals present in the solid form here. It is in the solid form, isn't it? It is in the solid form. Now, in the leaching process, what we are trying to do, we are trying to get this solid into the liquid form. So, we are trying to dissolve these heavy metals in acid. That is why you see that many in the leaching acid or some sort of or sometimes even base is used, any solution. Now, what kind of leaching solution will be used? In general, if you look at the electronic recovery, what kind of any leaching solution which has the potential of bringing the required metal, the targeted metal from the solid phase to the liquid phase. Whichever whether it is an acid or a base, does not matter, whichever serves the purpose of bringing these heavy metals from the solid phase to the liquid phase, that is kind of the leaching fluid will be used. So, here 1 to 6 molar HNO3. So, 1 to 6 molar HNO3 is used to get the things in solution. Some of them will come in solution, some of them may precipitate depending on the solubility product. Remember, you all have done that KSP. When IAP, ion activity product, is greater than KSP, when IAP ion activity product is greater than KSP, things will precipitate. That's, isn't it? We all learned about it in our high school chemistry. This concept is actually used a lot in environmental remediation, environmental this kind of exercise, chemistry, chemical industry. So, this concept of solubility product is it's a very basic concept which we use a lot in terms of our in terms of our application in different industrial process. So, here you will have tin precipitation as metastennic acid. So, tin will precipitate out. So, if the tin is precipitating out, so we can take it out in the solid phase. Now, you filter. After filtration, let us clean this so that it becomes clear. So, you have the tin precipitation. After that, you do the filtration. After filtration, now what we have? We have the tin is gone, most of it. There may be some is still left, but the tin is gone. Then we have the other, there could be other metals present, and little bit of tin and other things could be present as well. So, you dissolve in 1.5 molar HCl, dissolution is 1.5 molar. You could have some electrode deposition in tin, and neutralization by sodium hydroxide, that will have electrode deposition of copper and lead. So, you can recover copper and lead that way. Then you can use H2O regeneration for reuse. So, you try to reuse it back. HCl, so some with HNO3, some with HCl again, then we can recover HCl recovery for H2SNO3 solution. So, we get back over there. So, this whole, that is how the process is done. Here the target has been tin, copper and lead, but if the target has been something else, the type of solution may have changed. The type of solution that is needed and the pH conditions. So, basically we are playing with the chemistry of that heavy metal. If you have, we are essentially playing with the chemistry of that heavy metal and trying to separate them out. There is also a concept called sequential extraction. Now, what does that mean? Sequential means in sequence, isn't it? So, don't try to memorize, try to understand. Sequential will be what? In sequence. So, in sequence means you will try first one metal. Once that has been recovered, then you go to the another metal, then you go to the another metal, then you go to another metal. So, that way that is the sequence, you are following a particular sequence at extracting one metal at a time or one element at a time, depending on what you are looking at. So, that is what those are the concepts used in terms of recovery of metals from electronic waste. And here we are looking at an example of printed circuit board. Then so that's so far we have looked at several methods in terms of mostly based on chemistry. Now, we will be looking at some methods where we are chemistry is it is more as a biochemical method. Now, what we mean by biochemical, the biology is actually also playing a major role here, because if you think about the mother nature there are a lot of microbes, a lot of bio actually it is anything happening in the nature it can be biology, we could be there, chemistry could be there, geology could be there, a lot of other things could be there. Many times we call it like a biogeochemistry as well, because all these factors are there. So, and if you have taken any solid waste class, we talked about in the landfill or anaerobic digestion, or as simple as BOD test. BOD test is a biochemical test, isn't it? Because the bacteria are there, which is doing your most of the oxidation, and the oxygen is consumed because of that. But there could be some chemical reactions happening as well based on what kind of contaminants are present. There might be some reactions going on. But when we say COD, which is the chemical oxygen demand, which is essentially we are trying to make it chemistry test, like it's a chemistry test, very quickly that is done. So now we'll look at some of these biometallurgical process where we are making use of these biological methods. Biological methods in terms of metallurgical process means what? Mostly it will be leading to the extraction of metals. Whole this metallurgical engineering is based on trying to get, so mining people mines gets the raw material. Then our metallurgy people gets those from the raw material to its more usable form. Then the mechanical and the other people will try to have those use those material like aluminum sheets, copper sheets, which the material, and then also civil, electrical, and all. We will use all those material, and then now even computer science, hardware engineers. So we are actually indebted. If there is no good mining engineers, no good metallurgy engineers, we cannot really do much because we will not have the material. Even if you want to make a chip, these days there are so much fascination about all these electronics, data analytics, and financial engineering, financial risk management, whatever. But for anything to be built, we need material, we need energy, and for that, well, we have to really be very, very thankful to our mining people and metallurgy people, because they make things happen. For us, they prepare the raw material for us, which we can use in different applications, and material science and other people. So when I'm saying mining and the material, I'm talking about all people who are involved in those particular area. So let's look at the biometallurgical process. In terms of what is that? It's a biochemical process. It's involved in the treatment of metals. It has been using for last more than 2 decades. Actually, people are working on growing investigation for the last 20 years. It also there is another side of application. As an environmental engineer, we also talk about bioremediation of we try to do bioremediation, which is again we are trying to extract metal. This when we try to do the bioremediation, what we are trying to do, we are trying to get the metals, especially when we talk about this phytoremediation, for example. That is also one kind of mining only. You are trying to mine. You are trying to get this raw, like arsenic or whatever, cadmium or whatever the contaminant from the soil, which is the contaminated soil, to the biomass. So that's again one sort of reaction taking place there. So it's a material and mineral industry. There is great interest because it's a new technology, and it's a comparatively green technology. So one of the most promising technologies, that biometallurgical process. People want to use it to find out. At present, mostly research and development is in process. We can for copper, nickel, cobalt, zinc, gold, silver, and for all these we use microbes. Again, anything bio, you use microbes, isn't it? Microbes are used. So microbes are employed for a structural function and catalytic function. So we are using microbes, and they are both different types of microbes are there. You don't have to worry about the type of the microbe. You just try to understand the concept. And don't as an engineer, we don't have to remember the microbe's name and the type of microbes. Whenever we'll be designing this biometallurgical process, we'll get people involved from the biology side who can help us with that. So, but we need to, as an engineer, we need to understand like how this process will work. The process is more important, and the details of the process in terms of the more specific anything biology, we can have the biologist help us with that. So biometallurgy, you can have bioleaching and biosorption. Now what is bioleaching and biosorption? Leaching we already talked about, sorption we already talked about. So we have just added a term bio there, a small like a 3-letter word bio there. So, what is bioleaching means? Leaching with the help of biological organisms. So, you are trying to have things. What is leaching? Leaching is things moving from solid phase to the liquid phase. So, here earlier we use the chemicals, now here we use the microorganisms to do that work for you. Similarly, we can have the biosorption. It can be on the biofilm, it can be certain so like if things can sharp onto the biological so kind of surfaces, we can say, and we will talk about that in a little bit in more detail, and we make things happen that way. So, bioleaching it's successfully applied in many in recovery of metals from metallic sulfides. People have used it for metallic sulfides, which are the major bearing minerals for many base and precious metal. So, sulfides, why sulfides? Have you ever thought that I have just made a statement here that metallic sulfides are major bearing minerals for many base and precious metals. Why sulfides are major bearing mineral for many stuff? Why not anything else? Why sulfide? Because sulfide, typically if you remember from your KSP solubility product, sulfide has pretty low solubility product in most of the cases. So, for most of the metals. So, that makes it easy to be precipitated out. So, in the nature we have most of these minerals, most of these ores like where things are in sulfide form, copper or lead or arsenic, they are always in iron, they are in the form of sulfide because the solubility product is pretty low, things do get precipitate and it comes into the solid form. So, that is the bioleaching part where we try to recover metal from sulfides. Biosorption process is essentially a physico-chemical interaction where between the charged surface and charged surface of microorganisms. So, it is a charged surface of the groups of microorganisms and the ions in solution, and which livings as well as the dead organisms is used. So, you will have that is the process of biosorption, and we will talk about these two in little bit detail. So, in classification of bioleaching, you can have direct, indirect. Now, direct leaching where you are using like a biological organism, they are helping. So, how they will help in the leaching? They will break down those whatever is the big molecule present in which our targeted heavy metals is present. They will break it down in some like a reaction, and as a part of breaking down, the iron the metal will get free and it will come into the solution. So, that is what essentially it is going to happen. Indirect leaching, like sometimes using the thiosulfate pathway, polysulfide pathway, things do get formed. So, not directly it is going there, it is becoming from the sulfide, it is becoming sulfate. So, that is, and from the sulfate you can recover those heavy metals, or from polysulfide you can recover those heavy metals. So, that is the indirect. So, but things will ultimately come to the solution. So, in the metal sulfide action, if you look at the bioleaching direct, where you have microorganisms will directly oxidize minerals and solubilize metals. So, you have this metal sulfide. So, H2 in the presence of H2SO4, oxygen. So, it will make metal sulfate, which sulfate means it will come in solution. So, it will solubilize the metal and then sulfur will come out, and the sulfur can be used to get H2SO4, and that the reaction can go back. So, we can use more H2SO4. So, here it is a bacterial mediated reaction. So, it is the bacteria which is doing the job. So, it is a bacteria mediated reaction. So, bacteria is doing the work for us, and things are getting into the sulfide phase, and which is the soluble phase. Indirect method, what you will do, it's again you have your metal sulfide. So, in the case of like a ferric ion is the oxidizing agent. So, you have metal sulfide with the ferric ion. So, this will act, this will take a electron and becomes Fe2, and in that process the sulfur. So, here we have this will be an electron acceptor. So, electron will get released. So, so electron will come from actually here. So, your M2 plus and S. So, this will get sulfur, this bond will get broken down by the iron reducing bacteria, which will work actually for you and get M2 plus, and now this Fe2 plus you can oxidize it back and make Fe3, and then Fe3 can be used back into the reaction. So, here your iron reducing bacteria, many times you hear the term IRBs. IRBs are used to, they are reducing iron 3 to iron 2, and in that process when the iron 3 accepts an electron, so electron has to be come from somewhere. So, electron will come in the process of when Ms will broke down, and then M2 plus, and then 2 sulfur, it is negatively charged. So, the negative charge of that particular thing has to come out in terms of electron, and that electron will be taken up by iron to make iron 2 to go from iron 3 to iron 2. Now, iron 2 can be oxidized back to iron 3 and can be used back over here. So, that is the cycle can continue. So, metal sulfide here are leached via 2 different reactions. So, we can have via thiosulfate or polysulfide. So, those reactions are used as a intermediary reaction basically. Thiosulfate leaching is also done. Here you can have a thiosulfate mechanism where iron 3, so FeS2 where this will go to iron from iron 3 it will go to iron 2. In between they will have a thiosulfate being formed, and from thiosulfate then again iron 3 it will go to iron 2 plus. Iron 2 plus is soluble, iron 3 plus is non-soluble. So, it is a based on that you can get this iron out from this particular reaction. So, it is you can make more and more iron 2. Iron 2 is in solution, and you can from then you can recover iron 2 and make those iron products. So, polysulfide again the other way of doing it, do not worry too much about the mechanism. So, there are different ways of doing that. So, a polysulfide mechanism allows for a dissolution of iron 3 into by proton. So, in both leaching bacteria consist of regeneration of iron 3 ion. So, bacteria is making a use of in terms of iron 3 ions, the iron reducing bacteria is working here. Acidophilic iron 2 oxidizer, so controls the redox. There is a iron 3 iron 2 ratio is important. So, my main drivers are iron oxidizing microorganisms. So, there are iron reducing bacteria, there are iron oxidizing bacteria as well. So, we can this iron oxidizing bacteria can be used for that particular purpose, and let us see what happened here, just a sec. Yeah. So, we were yes. So, iron oxidizing microorganisms either in the mesophilic or the thermophilic range that can be used for that. So, from e-waste there has been some study being done, say dust were collected from the shredding process of e-scrap. The leaching experiment was performed using the bacteria. Do not worry too much about the type of bacteria, because it is basically it is a bacteria which can oxidize iron, and so the 2 steps were involved in reduce the toxic effects. So, first stage biomass was introduced in the absence of electronic scrap. So, first we want to make sure that if we load too much, what happens with the bacteria is that if they get inhibited, if there becomes toxic, it becomes a problem. So, we want to make sure that the bacteria survive. So, in that case we first make the life comfortable, make them acclimated to the atmosphere. So, biomass was produced in the absence of electronic scrap first, then was added in different concentration, and the cultures were incubated. So, leaching result confirmed that 5 to 10 grams per liter respectively. So, we did find and this thiobacilli were able to leach more than 90% of the available aluminum, copper, nickel and zinc. So, they could bring these into the solution. At higher concentration, the mobilization was reduced, maybe because it was becoming a little bit of toxicity, or it was getting toxic to those microorganisms. Lead and tin were not detected in the leachate, so lead could not come out. It was possibly lead become precipitated out, or and tin also got precipitated out for so it did not come into solution at that particular pH. So, for the copper for the printed copper from the printed circuit board, that also been looked into by some researchers. So, here iron sulfate formed using that bacteria, ferrooxidants oxidizes the element copper, and from PCBs to the cupric ion. So, those are the way it has been already kind of talked about. Biosorbents are also being used from for amendment like a waste biomass of algae, fungi, bacteria. So, they are used as they are at the surface. For the sorption you need a surface, isn't it? So, acid condition you can have biosorption of precious metals from solution. So, you can put it on the things can sorb on bacteria. So, research have looked at the previous metal, especially focused on gold. So, biomass may be used in this case as well. Biosorption by chitosan derivatives that has been used quite a bit, alternative for recovery of precious metal. Chitosan is a deacylated derivatives of chitin. So, that is where second most abundant like a polymer on earth after cellulose. So, major advantage of using chitosan is that amino sites of chitosans are easily protonated in acid media. So, it is a protonated, then it is a charged, and then you can use that charge to get the other charge that is attenuating the electrostatic force, often that increases the sorption. So, freely suspended biomass has some disadvantage because of small size and all that. So, in terms of the biosorption, what we are trying to do, we are trying to make a bacterial surface with a charge, so that the metals that we are trying to target will get attracted to this particular charge and shove into it, and then we can recover, we can take this biomass out and recover heavy metals from there. So, that is the pretty much the concept behind it. Okay. So, mechanism, it's as I was trying to explain, you get simplified as chemical sorption mechanism. Chemical sorption, you have the complexation, chelation, microprecipitation, microbial reduction. Physical sorption means generally electrostatic force and ion exchange, which we talked about. So, biosorption, it can be a chemical sorption mechanism as well as physical, could be combination. It is a kind of a hybrid. So, generally biological leaching is a cost effective technique, but it does take time. Anything biological take times, and complete recovery just by biological may not be enough. So, chemical leaching is comparatively rapid. So, many times you try to use it, but chemical leaching also has like a hazardous waste problem and all that. So, many times we try to use it in combination. Hybrid techniques have been proposed involving combination of chemicals and safer chemical biological leaching, and it can enhance the efficiency as well. So, hybrid technology using extraction of jet, tin, copper involving EDTA, using ferrooxidants, bacterial stain of DSM. So, all these, as you can see, several papers have been have looked into these type of hybrid technology. Not that much popular in industry right now, but things have been used from there. So, let us stop at this particular point, and we will continue our discussion in the next video, where we will start with this hybrid technology, and then we will talk about some of the e-waste issues, specifically for India, and that would be the last I think the last video for this particular week. So, again, thank you, looking forward to your discussion on the discussion forum. We will be more than happy to answer any question, and I hope you are enjoying the course.